Good evening, everyone. Today we will have a topic on cell growth, division, and differentiation and functions of the cells are concerned. And this is one of the important thing, very basically, to learn about how a cell divides and how we need to mitigate the cell division and what are all the parameters we need to observe during the uh, cell division is concerned. And hence, we are here to learn about this particular thing is concerned. Then the cell cycle and cell division is a process in which the every animal or the matter of the animal is concerned is going to be. So everyone know that the study and growth and differentiation undergone by an organisms involves the multiplication of so many cells. Then it's one of the most complex phenomenon that is especially on the living beings. So the particular chapter is consisting of the cell division, then importance of the cell division, the prokaryotic and eukaryotic cell division, cell cycle, regulation of the cell cycle. All these chapters will be dealt one by one. Then the objectives of this lesson is to define cell division and cell cycle, identify the purpose of the cell division, describe cell division in prokaryotic and eukaryotic cell, describe the cell cycle. Then cell division is the process by which the cell divides to form two new cells. So three types of the cell division are noticed. One is the prokaryotic cell division or the bacterial cell division, or it's also known as the binary fission cell division. Here, a cell divides forming two new identical cells, whereas the eukaryotic cells, like the human cells or the animal cells, we can say that it is consisting of the meiosis and also the mitosis. So the cell or the organism will grow by this method, and there will be a replacement or repair of the damaged cells which are already damaged. Whereas the meiosis is formation of sex cells or gamete cells. Then what is the question? Why do these particular cells divide? Is the cells divide for growth, development, repair, or worn out of the tissues, or to remove the old cells by a process known as apoptosis, and to also facilitate the exchange of the material and to control DNA overloading in a particular cell. These are all the objectives why a cell has to divide. And the prokaryotic cell division is very simple type of the cell division, and it always consists of binary fission. Then these three steps are DNA replication, then chromosome segregation, then cytokinesis. And each new daughter cell is genetically identical to the parent cells. Then the mother cell is a prokaryotic cell, is going to divide as cell elongates and replicates its DNA material which is inside this particular cell, and it is divided into two halves. Later, each cell is going to divide the genetic material among each, and the identical daughter cells are generated. Whereas the eukaryotic cell division is little more complex, and the cell division results in two daughter cells, each having the same number and kind of the chromosome as that of the parent cell, whereas the mitosis is consisting of two important phases. So, the mitosis is consisting of the four steps, namely prophase, metaphase, anaphase, and telophase. Whereas the mitosis also consists of the cytokinesis. So kinesis means the cytoplasm divides, forming two new daughter cells, and each daughter cell is genetically identical to the parent cell. Whereas the mitosis is consisting of the division of the mother cell into the DNA replication of the strands into two daughter cells, and the daughter cells are identical to that of the mother cells, and they share the DNA matter into equally into two cells. Then eukaryotic division is continued with the statement that results in four daughter cells. Then it is another process known as the meiosis. So here it consisting of two major steps. One is mitosis. Here it also contains the prophase, metaphase, anaphase, and telophase, and also the cell divides into the several cells, especially. So, cell cycle always is a process of generation of new cells and the non-existence of the old cells. Why it is there means it arises and divides into several daughter cells, and it also consists of the phaser growth and development, the secondly followed by DNA replication, thirdly preparation for cell division in larger scale, then actual happening of the cell division. So, cell after division begins once again a new cycle is concerned. Here you can see that the cell cycle consists of two main periods. Number one, it is the interphase. Interphase. Second one is the mitotic phase. So, the cell growth is also known as G1 phase. Then DNA replication is a phase which is called as the S phase, whereas preparation for mitosis is also noted as G2 protein, whereas the M phase consisting of the mitosis phase consisting of the cell divisions, especially the prophase, metaphase, anaphase, telophase, and the cytokinesis. So these are very much required to the cell is concerned. Then cell cycle, its interface is three types of the interface, because here the exchange of the DNA material between the daughter cells it happens. So it is the first phase is the G1 phase. Cell increases in size. Here also, in this phase, cell increases in size. Then in S phase, replication of the DNA. two sister strands of DNA are called as the chromatids, and these are produced. Whereas the G2 phase is the organelles double, then new cytoplasm forms all other structures needed for the mitosis form, and the daughter cell will be very equivalent to that of the mother cell, containing all the ingredients of the mother cell. Here there is a structure of the chromosome, and we have learned this in detail in the earlier class, like P arm, Q arm, etc. Then the end consisting of the DNA molecules which are adhering to both of these bodies. As you know, the cell cycle is already dealt. In cell cycle, the mitotic phase is the stage when cells actually divide and separate to one from the other. Whereas the mitosis is the division of the single nucleus into genetically identical daughter and also the nuclei. So, this division involves further two steps: division of the nucleus, because nucleus, once again, it has to divide among the newly produced cells, and separation of the cytoplasm and the new nuclei into the daughter cells is the next process. The mitotic phase is divided into two further phases, whereas the first mitotic phase contains four different stages. It's also called as very shortly known as P-MAT or PMAT. So it's also known as prophase, metaphase, anaphase, and telophase. Whereas the second metaphase is the mitotic phase is the cytokine cell containing the cytokine. So the phases of the mitosis can easily divide at IPMAT: interphase, prophase, metaphase, anaphase, and telophase. So in this you can see that the interphase is the single cell from which the cell division starts. So the cell structures are either drawn to the north pole or to the south pole, like that. Then in the prophase, all the arrangements about the requirement of different DNA materials and also the protein requirements will be met in locally. Whereas the prophase, in which the cell organelles starts division, then late metaphase also the several divisions are already happening. Then the mitosis is also known as PMAT, anaphase, telophase, prophase, and telophase. Then interphase is the one in which the centrioles, then nuclear membrane, then nucleolus, one of the important thing which controls nowadays the function of an entire cell. So early prophase, chromatid condense becoming chromosomes, nucleolus disappears, then centrioles separate and start moving to opposite ends of the cell. Then spindle begins to form, and centrioles, spindle made up of the microtubules. Then chromatids connected by the chromosomes. Then late prophase, in which you can see that on a cell the centrioles are going always away from the center, whereas the spindle is completely, it is formed at this particular stage, and which are all the required material to the growth and development of the animal. All these need to be stopped at once somewhere else. Whereas in case of metaphase, the chromosomes are aligned at the metaphase plate. Here you can see that there is a plate known as the chromosomes. They are all looking like this, like a band, and the metaphase happens in which matter the centrioles move at polar ends, means which attract the water. And in view of this thing, the centrioles might have moved at polar ends and projections as one of the best tools. But the spindle composed of the microtubules, they will try to drag the materials, genetic materials, etc., to their side. Then anaphase, the paired chromosomes separate, separated chromatids move to the opposite poles, opposite directions. Then partial division of the cytoplasm, now it becomes. Then telophase is the chromosome at the particularly the poles, means end of a particular life of a cell. Chromosomes uncoil to turn chromatin nuclear envelopes of the particular nucleus it develops. Then spindle it to the fiber, it disappears, and cytokines out occurs at the end of the mitosis. So animals have a cleavage outflow for separate daughter cells, whereas the plant cell, a cell plate repatriates the daughter cells, whereas the daughter cells are genetically identical. So usually always cellulose return to interface. So the mitosis, we can get lot of animations, etc. So we can find the source. So by this, I am going to thank you for being patient with this thing, and thanks a lot. Bye.